Okay, so I'm going to introduce our next speaker. Our next speaker is Dr. Bilal Chami, and he's got a massive bio, another person with a huge bio. He's an academic researcher. I think you are all going to absolutely love this session. It's going to be filled with lots of interesting content. He's won many awards. Please put your hands together for this fabulous Dr. Bilal Chami. Thank you very much. And he's going to be presenting antioxidants, which we all know, we all need, and they're fabulous. Thank you so much. Thank you, Doctor.
Thank you, MedTech, for the invitation to speak and for the very flattering and overindulgent introduction. My name is Dr. Bilal Chami. I am an academic researcher, and I think I'm a bit of a fish out of water here. And that's good because I've just been here for the first time at one of these conferences. It gives me a really good insight into a different area that, and I can draw a lot of parallels between what's happening with all the technology that we're using, all the different talks about different biostimulators and what they're doing to the cell. But the aim of my talk, let's go back. Might just use that, yeah. So the aim of my talk is to introduce what antioxidants do to our skin, but I think we really need to re-understand what aging means. And so I think at the end of my session, if I've convinced you to think about aging a bit differently, then I've succeeded. So I'm going to try, and just bear with me. But also, not just the aging process, where do antioxidants come into this place? And how does it all tie together? So we all think of aging in this way here, and some of you might even have these diagrams blown up into your offices. We think of it, at least superficially, that there is all these conditions occurring. There's fine lines, deep lines. There are changes to the facial structures, even changes to actually the orbital bone resorption, causing deeper eye sinking, loss of the jawline. And we can go a bit further. We can look at schematics. Now, schematics are useful because we can break down, look into the histology, and find that actually there is changes to the cells here. The density of cells are different, and also the changes to the overall thickness is present. But I would like to think of it a bit differently, actually. I think I would encourage you to think about aging in two ways. There is chronological aging, and there's not much you can do about that. And that is just the age which you were born. So if you're 40 years old, well, then you've lived on this earth for 40 years. But then there is biological aging. Now, those two are related, but they're not the same thing. So you could be 40, 50, 60 years old, but it doesn't mean that the biological age reflects that. We think of aging as being some sort of linearity. So that we are, I'm just going to step down the stage here so I can see the screen a bit better, that if our biological age should match our chronological age. So if we are 40 years old by chronological age, then our biological age is about 40 as well. But that's not true. We know that various lifestyle changes that we have or that we've adapted can actually accelerate our aging. But at the same token, we can also slow the aging process by the exact same virtue of changing the way which we live. That is not exposing ourselves to certain age promoters. So the real way to think of this is, rather than how old we are, is how old are our cells. And this is a new area of science that we're getting better and better at. So much so that there are technologies that exist now that can actually age your cells, independent of how old you are. So independent of your chronological age, we can determine the age of your cells. And it is really dependent on many lifestyle factors and environmental stresses and exposures to sun. This is a cell type that this audience needs no introduction to, I don't think, dermal fibroblasts. But just a quick recap, these cells are the major cell type that are present in the dermis, and they have a really good function. They're actually responsible for developing all that connective tissue. So they make collagen, but they also make elastic fibers. They make glycosaminoglycans, or otherwise known as the G molecule, and they make things like hyaluronic acid. And these are incredibly mobile cells. With fibroblasts, they zip around. And one of my postdocs, we used to use a microscope to watch and to track fibroblasts. And very often we had to change the position of our microscope just to keep up with how fast these fibroblasts actually move on stage. So they're very, very quick. And they need to be because they're really important for wound healing. Now, I need you to bear that in mind how quick they are, because that's a critical part of the aging process. But coming back to what they do, so they lay down the basis of collagen and other interesting matrices, but they also resorb. So while they lay down collagen, they have to also degrade old collagen. And it's that balance that maintains the normal skin structure. They have to lay down as much as they degrade. And if they lay down too much collagen and they don't degrade enough collagen, well then, that's scar tissue. And if the opposite occurs, where they resorb a lot of collagen, and they don't make enough collagen, well then that's the fundamental process of aging. And if we look at them, and this is really curious because we all know what aging looks like, at least at the surface, but we don't know what aging cells look like. And here you see these fibroblasts. They're very young, and they're very slender, and they're very thin, and it's quite a normal appearance. There are many more numerous here, so there are many more fibroblasts here. These are fibroblasts that have aged, and what you'll note is that they don't look so long and slim anymore. They're actually shorter, they're a bit more stubby, and they've collected a lot of fat tissue as well. You know, you'd be surprised cells collect fat. And what that means for cells is that they're actually a little bit unwell. And they're actually quite sluggish. They don't move around so much. They don't divide as much. They're not very active. So they're not actually laying down a lot of collagen, but they're still resorbing collagen. And here's a slide here. I'm not sure if you can see that very well, but it's young tissue that's been cut. And all this little whirly pink stuff that you're looking at is collagen. And under an this is a scanning electron microscope, you can see collagen laid down in rope-like structures. And it's nice, it's quite organized, and it's very thin and slender. And this is what normal collagen laid down should look like. But in aged skin, it looks very different. You would see that the collagen here is not very densely packed. It's quite loose. The fibroblasts themselves look different. They look a bit more shorter and stubbier, just how they look here. And the way in which they lay down collagen is very disorganized. So now we have an idea of what old cells look like and how that affects the skin. But the real question that scientists ask themselves are, why does this happen to cells when they age? Why do cells become slower? Why do they not divide so frequently? And why does their activity decrease? And it all has to do with DNA damage. And this is really the frontier of aging. It's the DNA damage that occurs, but it's the accumulation of DNA damage, which is the real problem. And so if we think about this as being the leading cause of aging, then we should think about how we can mitigate that and how we can remove that as an aspect of our aging process to overall achieve better health. And better general health means better skin health as well. Now, I always look at what happens, particular syndromes, and because they give us the best understanding of what the aging process is. And this is a syndrome, these are called progeria syndromes, the syndromes of premature aging. And it occurs in this particular syndrome called Hutchinson-Gilford syndrome. It's a very rare syndrome. 1 in 20 million children are affected. This is a 4-year-old child, a normal child, and you can appreciate that looks quite normal, and a child affected by Hutchinson-Gilford progeria. And this child, although he is 4 years old, has biologically aged to about the age of 80. And you could see that he has all those features that we're familiar with, the thinning of the skin, the loss of the jaw, the increasing increased absorption of the orbital sockets there. And if you screen their DNA, and we have done this, we've noticed that there is a defect in a protein that causes the DNA to repair. So this child has a defect in the ability to repair its DNA. And that is what's driving the aging process. It's accelerating the process. So this is one example. I put at the very bottom that SIRT1 is a regulator of this protein that has been malformed. Now just keep that in mind because I'm going to end on this at the very end of the presentation. Here's another syndrome. This is an adult onset premature aging condition called Werner syndrome. Again, there's a defect in these individuals with their ability to repair their DNA. But they largely live normal lives up until the age of 20. And after the age of 20, they rapidly age. And this individual has an age of 56. So her chronological age is 56. Her biological age is around 80. So again, more evidence to show that your chronological age is not the same as your biological age. You could actually accelerate it. And later, I'll show you how you can decelerate it, how you can slow that process down. Again, SIRT1, this is really the key to today's talk, is a regulator of this protein that causes DNA repair. So, in summary, what have these premature aging conditions taught us? Well, they've taught us that DNA integrity and the repair of DNA is central. It really is central to slowing down aging. And how does DNA become damaged? That's the next question we ask ourselves. Well, it's really simple. There are ways in which we can damage it through our lifestyle, through stresses, exposure to sun, exposure to carcinogens, or to radiotherapies, or to chemotherapies, or chemicals. There are a lot of ways in which DNA can be damaged. But DNA can be damaged internally as well. I'll talk a bit about that soon. But it's the accumulation of that damage. The damage itself isn't always the problem. It's the accumulation of damage over many generations of cells that can lead down this pathway. The worst pathway is that it can cause cells to become cancerous. So all cancers come because those cells have become damaged through their DNA. But in most cases, those cells actually accumulate the damage and then they don't function so well. Cells that have a lot of damage tend to be more slower, tend to be more what we call senescent. They just hang around there, they don't do a lot. Every now and then they might divide, but they don't move much. And so, and this is kind of the end stage of aging. But you could see that it really does occur because of the accumulation of DNA damage. And over here, right where this red little blunt arrow is DNA repair. This is where we intervene. So it's all about DNA repair. And there are many ways where DNA can be damaged, and it's not so important how it's damaged, really. But what is important, or the way in which it's damaged, of course, we can change our lifestyles so that we minimize the way in which it's damaged. But what's really important is that we have a process that we upregulate to repair DNA as it's damaged. And I came across a little saying that I like so much that I thought I'd dedicate an entire slide towards. It is the imperfectness of all biological processes that leads to the accumulation of damage. So what this really means is that even if you've done everything right, it is the very virtue that you're sitting here breathing that is causing damage to your body. In other words, there's a trade-off to living. So the more longer you live, the more damage you will occur, even if you've done everything you could to mitigate it. So it's not, and it sounds all very dismal, and I promise I'm gonna flip it around. So there's fear and then hopefully a solution. But not to say that we shouldn't do things that will minimize our damage, but there are ways in which we can intervene. But again, sometimes damage just occurs because we've just lived long enough. Okay, and because it's not just about the damage, it's about the DNA repair, there is a two-pronged strategy that scientists have devised. The first is the obvious one, just to prevent damage to begin with in the first place. And this is lifestyle factors, avoiding sun exposure, avoiding exposure to UV, gamma radiations or whatnot, and chemicals that we know that cause DNA damage. That's really important. Antioxidants are really important because they mop up a lot of oxidative stress that we experience. I'm not going to dwell too much on that aspect. I think I might just end at that point. But we need to have a way to repair the damage that occurs. Because if you remember, it's no fault of your own, just the passage of time that will cause damage anyway. So increasing DNA repair mechanisms is essential. So back at this point here, and I hope I haven't lost you at this point, I promise it doesn't get more complicated than this, but sirtuins are a protein that we're really interested in the anti-aging community. And I'll introduce you to these proteins. These are a fascinating group of proteins. There are about 7 of them. And what they do is that they sense when cells are stressed. And I really couldn't help the analogies that Dr. Gregorius was drawing with all these technologies and how they induce some sort of stress to the dermis, epidermis, and how I've noticed that, and I want to come back to this at some point, that low-level stress for cells is a really good thing. But I'll come back to that point later. So, sirtuins are proteins that increase our DNA repair in our body. And because they increase DNA repair, they're linked to healthy aging. So simply put, the more sirtuins you have, the better your health and lifespan will be. And there's a lot of research that backs this. But just to put it into a bit of context is here, let's start off with DNA damage. It's happened because of no fault of your own, or possibly a little bit of a fault of your own. And sometimes what can happen is that those cells can accumulate the damage, and then they just kind of get a bit old and they stop dividing. In the worst case scenario, they die. But if you have increased levels of sirtuins, then the cells will repair themselves. And then that promotes the cell to survive, and the cell retains its youthfulness. So the take-home message for that slide is that sirtuins are really good for DNA repair. And the question you should be asking at this point, well, how do we increase our sirtuins? How do we do this? But I think the first real question is, what is the normal level of sirtuins that we have in our body? And the highest levels that we express are when we were fetuses. And as we age, it declines. And it doesn't decline in a way that is like nice and linear and slow. It actually occurs in 2 different dips. In our 20s, we experience the first sharp dip. And then again, we experience the second sharp dip in our 40s. And these are incidentally ages where we see the phenomena of aging occurring. The first set in our 20s at some point, and the second set we experience a major dip in sirtuins in our 40s. Now coming back to fibroblasts, because this is, I think, the cell type that we're all really interested in maintaining skin health. When they're aged, just to recap, they start to slow down, they don't divide so much, and they start to dissolve more collagen than they make. And that causes a whole bunch of changes to our skin. But when we make fibroblasts express more sirtuins, they actually rescue their phenotype. What that really means is that the fibroblasts begin to become more active. They start to move around, they zip around, they divide, they produce more collagen. And they're so strongly related to the youthfulness of fibroblasts that the cosmetic industry, and a paper that was released in 2022, was calling out for sirtuins to be a marker of the usefulness of active ingredients in the cosmetic industry. So it is so fundamentally tied to how youthful fibroblasts are that we really just can't look at this as an offset. It really is a marker of how healthy our skin is. And another slide I really couldn't pass up, because we know that sun damage is the worst thing that you could do to your skin. A 2011 paper summarized the impact of the sun on our skin. And about 90% of premature aging is caused by the cumulative sun exposure. Okay, so the best thing you can do for your skin is to wear some sort of sunblock, at least initially. But where this is really interesting is that in a study in 2014, they've taken biopsies from patients, and this is an example, where they've taken a skin biopsy of an area where there is sun exposure, so on their arms or the face, and then skin biopsies of areas that weren't exposed to the sun. And what they've shown is that sirtuin expression is highest in areas where there is no sun exposure. And that is usually where the skin looks very youthful. And areas that there is a high amount of sun exposure and where their skin looks more aged, there is low levels of sirtuins. So this really means that the sun is doing sun exposure is doing two things. One, it's actually directly damaging DNA. But not only is it damaging DNA, it's preventing DNA from being repaired. So it's a double whammy. Okay? It's damaging DNA on one side, and it's preventing that damage from being repaired later on. So really, you could see the impact of the sun that it has on our skin. And I thought that was a really interesting slide. It really summarizes the impact of sun and how it relates to these sirtuins. Okay, now this is the more positive note. So I just gave you a bit of a sigh. It's like, it's helpless. What can we do? There are things we can do. And there's a lot of research in this area. And I'm really delighted to kind of present this later, more uplifting side of the talk. Calorie restriction. This is not really the uplifting part, I promise. But the best way to induce, or not the best way, one way in which we can increase our sirtuins is by dieting. Okay? And well, look, it does something similar. It induces what we call metabolic low-level stress. And cells can exist in three areas. One, they can exist in a more comfortable area where they're just using glucose, it's making energy, it's doing all the things it's supposed to do. And it can exist in a high-stress environment. And if the cells exist in a high-stress environment for too long, they eventually die. It's the low-level stress is where the cells increase their defenses. And it actually promotes cells to survive. And it promotes the youthfulness of cells. So cells need to be put into this, kind of transient balance between normal activity and a low-level stress and bouncing back and forth. It's that bouncing back and forth that really makes those cells more resilient. So, 14, 16-hour fast, it causes our cells to switch over energy sources, which we use glucose. So we consume sugars, and sugar fuels our cells. If we fast for 14 hours, we've depleted a lot of our sugar. And so the cells are forced to use fat as an energy source. And when it switches from sugar to fat, it increases sirtuins. And when sirtuins increase, the fibroblasts get into action, they start to zip around a lot faster, they start to make new collagen, and they appear more healthy. So it's all a good story, except that it's very hard to prescribe dieting or calorie restriction, and so therefore this has got a very poor adherence. And this kind of birthed a new area of research. Well, is there a pill that we can take that will increase sirtuins? Yeah, and it's coming. And it's called the sirtuin activating compounds, or otherwise known as STACs. And resveratrol, and this is another one I wanted to hang on to. Resveratrol is one of the most potent SR or SIRT1 activators. There's a list here that I've placed. It's not a very large list. It's a bit bigger than this. On the top-hand side are naturally occurring antioxidants. On the bottom are some things that we've made in the lab. And they all, to some various degrees, activate sirtuins. But the best activator is the naturally occurring resveratrol. Has anybody heard of resveratrol? Fantastic. So this is going to be all positive news. Okay, but just really to impact and to highlight what are the effects of increasing sirtuins. This is a monkey or two monkeys over a very long experiment that was done. And these two monkeys lived to the age of 27. Now, 27 years in human years is about 86 monkey years. And one monkey was able to eat what we call ad libitum, so whenever it felt like it, pretty much the way we eat out and through our lives. And it's reached the age of 86. It's lost a lot of its fur. It's got this kind of hunched over gait. You can actually see the signs of aging on the face of the monkey: sunken eyes, bit of droopiness. The filum is actually quite long, and the lips have lost their volume. Now, the monkey on the right-hand side was only allowed to eat between 4 to 6 hours a day for 27 years. Now, no one can prescribe this, of course, but this is just a proof of concept that this monkey seems and is a lot more youthful, even at the age of 86. its tail is upright, it's maintained a lot of its fur, it's maintained the color of its fur as well. And even at the facial level, you could see that it hasn't lost a lot of orbital bone. And so you don't see a lot of sunken eyes or the associated puffiness under the eye. Its lips are more puffy. And so generally the skin, it's in a much better condition. So we know that this monkey lived a very healthy life, but we can't say that it lived a very happy life. And so, again, we're not prescribing this, but this is just a proof of concept that sirtuins are really important for healthy aging. And back to resveratrol, the most potent activator of sirtuins. And I've put a picture here of grapes because although it wasn't first isolated in grapes, somehow the wine community jumped on this. And I'm going to show you why it's, well, yes, but I'm going to show you why you can't get enough of it through wine. But it was first identified in 1939 by a Japanese scientist from a herb actually called Resveratrol album. And like most compounds, once they're discovered, we kind of forget about it. And in 2003, it was rediscovered by another scientist, David Sinclair, who's become a professor and a leading authority of anti-aging at Harvard University. And he showed that resveratrol had such potent anti-aging effects that it was one of the most potent upregulators of sirtuins. And since then, we've learned a lot more about resveratrol, where it comes from. Again, you know, red grapes is one source, not the only source, but it's one source. It's not a good idea to try to get resveratrol from wine. If you're trying to achieve the therapeutic dose, which is 500 milligrams, you would need to drink 40 liters of red wine a day. So any benefit you would get from resveratrol in your wine, you would have offset it by the liver damage that you would experience later on. Dr. Mike had this really interesting word called biostimulator. And I've never heard of that word used, and I thought it was actually a fantastic way to describe resveratrol. It really is a biostimulator, so I'll place that just there right before I thought I'll jump on and give my talk. So, how does it all work? Resveratrol mimics calorie restriction. So when you can't calorie restrict yourself, resveratrol is really the next contender. Because it acts on the exact same pathway as fasting and caloric restriction and exercise as well. It increases sirtuins, it promotes all these positive effects in our bodies. It does something else as well. And this is another concept. So putting DNA aside, cells ultimately make protein and they make fat. It's what cells do. But every time a cell makes a protein, that protein that it makes has a short lifespan. And it needs to be recycled. And the process of recycling, old protein, old fat, is called autophagy. And the cells need to enter a state of recycling in order to perform properly. Otherwise, you get toxic proteins building up and the cell becomes very, very sick and it dies. So taking resveratrol is also shown to increase the recycling of protein. So it's helping our DNA repair, it's recycling our protein, it's doing a lot of positive things. The question is, how do we take resveratrol to achieve all these effects? And this is where the science becomes tricky, and we're still kind of evolving. Hopefully I have enough time. Okay, fantastic. There is the oral route and there's the topical route. I'm going to convince you why the topical route is much more better way to take resveratrol. Oral is very convenient. It's just a pill a day. You walk away and you're done. And there are probably some gut microbiome interactions when you take resveratrol orally. It does get absorbed rather well. It's a fatty kind of compound, but the problem is with resveratrol, it's got very poor bioavailability. And what that means is that when you take it orally, because it goes through first pass metabolism, it goes straight to the liver and it gets broken down, it gets cleared really quickly. So it doesn't really hang around in the blood long enough to impart a good effect, and not nearly at the concentration that you need. So the science has kind of figured out, well, okay, we need X amount of resveratrol on cells for X amount of time to have an effect. And trying to take it orally is just really difficult to get that. And you kind of get a generalized distribution around the body. And there's some meaningful effects, but when we think of in the context of skin, it's quite difficult. The pros to using topical resveratrol as a serum is that it penetrates the skin very easily. It's very fatty, so that means that it gets deep. We have evidence to show that it actually accumulates in the skin and actually can even enter the bloodstream from the skin and have those systemic effects as well. So it's pretty good. There are two major problems, however. Poor water solubility, so it's really hard to get into a solution. And this process called rapid isomerization. I was tempted to not present this, and I thought actually it's really important part of science. I can't leave it out, so I'm going to try to explain this. But resveratrol, and this is what the molecule looks like, but it also looks like this. It's exactly the same molecule. The only difference is that it's actually bent out of shape. And that bending out of shape is a problem, because we know that resveratrol only works when it's only a good activator of sirtuins when it's in this conformation. So this is a summary of why topical is a preferred method of delivery. The scientific literature really has conflicting results on oral resveratrol and skin health. It's probably because we can't get enough of it, or at high enough concentrations at least. But topically, we know that actually it works a lot better than oral. So there's a study done, I think this is a 2014 study, showing that resveratrol, when applied topically, outperforms resveratrol when it's applied, or rather consumed orally. So coming back to this process here called called isomerization. These two are two naturally occurring forms of resveratrol. They both have the same antioxidant levels, but only one is an activator of sirtuin. And this is called a lock and key mechanism. So what we're looking at here is the protein sirtuins, so the protein that we're trying to activate. And resveratrol kind of fits into this pocket like a lock and key mechanism. You can see the molecule there, and that activates the protein. But it only works when resveratrol is in this formation. If it bends out of shape, it no longer fits properly in the protein. And so although you get the antioxidant effect, you don't actually get the sirtuin upregulating effect. And to be honest, it's a pretty lousy antioxidant. We have more potent antioxidants. So it's not acting by virtue of its antioxidant ability, but rather it's working as a drug, you could think of. So this is the form that you should be looking for when you're reading the back of your serums. Is it trans resveratrol? So back to the problem. It's poorly water soluble, so it's really hard to formulate. It does this thing where if you eventually put it into formulation, it converts by itself to the form that is pretty much useless as a sirtuin activator. So what do? Well, we use something called cyclodextrins. And Grigoris pointed out that knowledge not shared is useless. And although this is knowledge that we've acquired, we think we still should be obliged to share it. Cyclodextrins are naturally, or some of them are naturally occurring. These are sugar molecules that look like baskets, or a hole in a basket. And we use it in food, and we use it in many different industries. And they're very, very safe. The FDA has given it the highest status of safety called GRAS. So it's got this GRAS status. And they look like this. There are many different forms, but every form has just got a bigger basket. And your drug fits into that basket. And I'll show you why that really works very well. When you use resveratrol, all of a sudden, the problem of solubility goes out the window. You can solubilize almost anything. It makes your product or your drug more stable, and it increases the bioavailability. Okay. So how it works, this is your cyclodextrin. This is the drug that you're trying to solubilize and hopefully prevent from isomerizing. It simply fits into, and it creates this kind of bucket apparatus. And when it's done on both ends, you can see that the drug now is protected by two buckets of sugar molecules. And it prevents it from bending. And so now you have a solution to your problem. You can solubilize resveratrol, and you can keep it in its trans resveratrol form, the useful form that you need for your skin. Oh, and I'm almost at the end of my talk, so I'll wrap up pretty quickly, I think. Okay, so what is the ideal resveratrol complex percentage and what is the ideal way to formulate? Most of the studies that have been done looked at 0.25 to 1% with the best efficacious concentrations around the 0.5 to 1%. Resveratrol works best when mixed with other antioxidants, and this is tapping into something called antioxidant synergy. So in other words, when two antioxidants are used independently, they have their own activity, and when you use them together, you don't get a double up, you get like a triple up. So this is a case where 1 + 1 is 4. And this is a graph showing you exactly that. This is actually resveratrol. What you're looking at is the antioxidant ability or the activity. And resveratrol is actually down the bottom there. Quercetin is another type of antioxidant that occurs naturally. And it's got a high antioxidant activity, but it's not a very great activator of SIRT1s. But when you use these two together, you get a 1 + 1 is 3. And so the antioxidant capacity increases a lot better. So again, higher concentrations, best formulated with other types of polyphenols as well. Quality matters. In our R&D that we've done for many different suppliers, you get resveratrol looking like many different powders. And when we've done our own HPLC mass spec analysis, we found that only one form of, we can see almost with our eyes, gives you the best quality of resveratrol. And some of this is just kind of either rancid or bulked up. So I thought I'd just throw that in there. Okay, so hopefully I've convinced you all the way in which I think we should think of aging, and this is really echoing where the scientific understanding of aging is, that we need to have quality DNA. And quality DNA starts from minimizing damage where we can, but also repairing it as well where we can as well. And the accumulation of damage is really the principal driver of aging. SIRT1 is the protein responsible for repairing DNA and kickstarting the process of DNA repair. And resveratrol is a natural antioxidant that promotes DNA repair. It's the most potent activator of SIRT1s. And topical resveratrol is likely more superior than oral resveratrol, but it just needs to be really well formulated. So that brings me to the end of my talk. I think I'm just about on time. A bit of background about how we are. I'm a biologist, and so somehow I found my way into this arena of skincare, and it's been a fascinating journey. We were conceived in 2021. The concept, we quickly realized that we weren't able to do anything unique unless we started our own research facility, and that's when we established our research facility and performed the R&D from 2001 to 2023 when we launched. Our ethos is really bringing science back into skincare, and that starts from having a really good understanding of what causes the conditions that we are trying to treat. And so we have a team of scientists that work on this as well. We're trying to simplify the whole process of topical skincare and make what we call multiplex skincare. So this is looking at antioxidant synergy, picking particular kinds of combinations of antioxidants that work together to give the best antioxidant effects on our skin. And our B2B model, where we're working with clinics to customize serums for their particular needs. I think we look at clinicians as being the ultimate observers to their patients, and they often have really good ideas of what active ingredients work for their clients. And so we're kind of tapping into how we could assist them by formulating specifically for their particular needs.